Good morning and welcome back to the Mistra SafeChem final webinar for phase 1. We are now starting up session 2 this morning. That's with the title Catalysis for Sustainable Organic Chemistry and Upcycling of Forestry and Textile Residues. The morning agenda looks like this. We have finished session 1 and we are now moving into session 2, where we have a number of speakers who will present results from Mistra SafeChem in phase 1. You see all their names and titles here. And I will want to remind you that we have at the end a 10-minute Q&A session, and you are warmly welcome to send in your questions in the chat function, and we will compile and ask them towards the end of the session. With that, I will give the floor to the first speaker of this session, Belén Martin-Matute. Please, go ahead. Thank you.
Good morning, everybody. So my name is Belén Martin-Matute from Stockholm University, and I will be introducing the work in Mistra SafeChem phase 1 coming from Work Package 4. So the aims and the goals of Work Package 4 has been the development of cutting-edge green chemistry processes and methods aiming at significant reducing toxic waste and hazardous byproducts associated with current chemical synthesis and processing employed in the industry. We have also worked a lot on using bioresources for the synthesis of chemicals, and you will get today an overview of all these aspects. So in the area of developing new methods for organic synthesis, it's been primarily Professor Bäckvall, myself, and Professor Syrén. And then on the area of the use of different bioresources will be Mathew and Joseph Samek, both of them professors also at the Stockholm University. So you will hear from them or from their students on the research performed. Yes, so we all know we are dependent on making chemicals, and the special areas that we have been focused on have been in the healthcare and pharmaceutical synthesis, fertilizers and agrochemicals, and also new materials for technological applications. So we need chemicals, and chemicals need to be produced, but we need to produce them in a sustainable manner. And in this picture, catalysis plays a very important role. So when we want to make an organic compound, particularly when it's a very complex organic compound, we may develop a catalyst that finds an alternative reaction pathway that makes possible that transformation in a selective manner that also involves to reduce the number of byproducts and also to have high yields of the desired product. Without the catalyst, perhaps the reaction does not even happen. So developing new catalytic processes is very important to access these important compounds, as I discussed in the previous slide, especially in the area of the pharmaceutical industry. So within Work Package 4, in the area of catalysis, in the first few minutes, we will discuss some replacement of noble metals by abundant metals to produce organic compounds. We have also worked on the use of carbon dioxide as a one-carbon syntone for synthetic organic chemistry. We have also worked a lot on developing new heterogeneous catalysts for recovering the metals, and at the same time as producing complex organic molecules. And we have also worked a lot on the use and developing of new catalysts for the synthesis of pharmaceuticals and new drug candidates via late-stage functionalizations. We will primarily focus on the 3 points, but I can briefly say that this last point deals with developing a catalyst that can modify a pharmaceutical in a very selective manner, in a way that in a single step you can get a new potential drug candidate. And this saves a lot of time, a lot of resources, and it really helps to move very quick in the discovery of new pharmaceuticals. So first, I will now go into the replacement of noble metals. And one of the reactions we've focused on is the hydrogenation of unsaturated carbon-carbon bonds. Here I show a triple bond that is reduced selectively to a cis alkene or Z alkene. And traditionally, this has been done using a palladium catalyst, which is a noble metal and is not very abundant and also is pyrophoric. And as the reducing agent, traditionally, the industry use dihydrogen, which is also obtained from fossil fuels and is also highly flammable. This reaction per se is very important in many processes, and here are some important compounds that are produced by reducing a triple bond, many of them with important anti-tumor activity or also in the industry of fatty acids, etc. So providing another alternative method for this hydrogenation is very important. So here what we have done was to replace the palladium catalyst by abundant nickel catalyst. And this is a nickel foam. And here you have a picture of it. And we have also worked on replacing dihydrogen by acidic water. So in order to use this, then we need to use also electricity. So this is an electrochemical reduction. So we started investigating this reaction, and this is some of the scope that we achieve, going from triple bonds to double bonds selectively. This is some of the scope, and what we can see here is that the yields are very good. The reaction also tolerates many functional groups, and also what is good is that it tolerates some functional groups that are not compatible with the traditional palladium catalyst. Then another advantage of this method is just by replacing water by deuterated water, which is also highly available, we have also access to deuterated olefins, and these are also highly important in medicinal chemistry. So the reaction is very efficient. Our next step here was to try to develop a similar method that couldn't need the use of palladium to reduce the double bond to fully saturated organic compounds. And this is what we show here. And now when we took up this study, what we really wanted to show is, or more to compare, what are really the advantages of this method compared to the traditional one in terms of sustainability. So this is the setup that we used, is the electrochemical setup, where we have here the working electrode, that for us is the nickel foam, and we have a counter electrode, which is a platinum wire, and then we of course need a reference electrode. And we optimized the reaction, studied the scope, as I will show just in a minute. Important for us is to show that the metals used are recyclable. And then what we thought it was extremely important here was to develop the method in collaboration with the other work packages within Mistra SafeChem to really make good choices regarding the reagents and also to screen all the byproducts that may be produced. So this was done in parallel, and I will give a few more details about this. So this is part of the scope. So you can reduce many double bonds to saturated compounds. We have also compatibility with many functional groups, such as amines or anilines. So the reaction works well, and it has a good scope that may be comparable to the palladium on carbon. Is the catalyst now recyclable? So that we did in this study. And what we do here, usually we look at low conversions, because what is very important is to show that the catalyst has the same reaction rate for every run that we test. And indeed, this is what it shows, this graph. We have a very good and very similar conversion, at least for during 15 recycling cycles. So the catalyst is very stable. This is for those that are interested, a little bit more detail of how the reaction happens. So the protons react with the nickel foam and 2 electrons, and you form these nickel hydrides, and these nickel hydrides are transferred to the unsaturated carbon-carbon bond, providing the final saturated compound. And then in the anode, we have the oxygen evolution reaction. So when we were developing this, we early on discovered that instead of using methanol as a solvent, we should change that for ethanol because methanol has intrinsic toxicity. And we were also using hydrochloric acid as the proton source, but that we immediately realized that this would produce chlorine, and that has some toxical concerns, and therefore we replaced that by sulfuric acid. We also did a non-targeted screening for this specific reaction and look at what type of byproducts are formed after the hydrogenation reaction, and none of them showed any significant toxicity. So that was good. But is it really better, this system, than the palladium in terms of toxicology and life cycle assessment? So that's what we did in collaboration also with the other work packages. And what we look into was into the electricity use. Well, I can go here, but better. We look into acidification, climate change, eutrophication in freshwater, eutrophication in marine water, resource use, ecotoxicity, and human toxicity, cancer and non-cancer. And what we can see from this graph in short is that the nickel has major concerns than the palladium system. And this is particularly the case in the resource use and acidification, being 50 and 14 times larger for the nickel system. For the rest of the impact categories, the difference is not so large. But what we can also see here is that these big differences are primarily produced by the use of the platinum counter electrode. So replacement of the counter electrode would make this nickel system very similar to the traditional palladium on carbon. And this can be done, and we can use a carbon counter electrode, and then these impact categories are not so affected in the nickel system. The other aspect we look into was at the electricity used in the nickel system. And here we made a prediction of the electricity scenario in year 2030. And what it can be seen here is that all the impact categories diminished, particularly they diminished more for the palladium on carbon. So this was our reduction catalyst, and now I will give the word to Professor Bäckvall, and he will discuss on the recyclability of metal catalysts.
Yes, I'm Jan-Erling Bäckvall from Stockholm University and also working at the Department of Organic Chemistry. All right, so we have been using, in the first part of my talk, we've been using crystalline cellulose as a carrier in sustainable catalytic organic reactions. And the crystalline cellulose as carrier has been used for palladium and copper, and we have developed carbocyclization and cross couplings. So in this first example, we have been using nanocellulose as a carrier for palladium, and in this reaction, aniline is used, and this is activated by the palladium to give this dianyl compound. And the question is, can we now react with carbon monoxide? Because carbon monoxide is a very useful reagent since it can be generated from carbon dioxide via reduction, as also discussed previously. And then we could get pyrrolidines. In the first attempt for this reaction, and I should mention that the linker AMP, aminopropyl, is used to tie palladium to the nanocrystalline cellulose. So in the first attempt, we got no yields. But look here what happened if we add just 1 mole% of silver triflate, that is twice the amount of the palladium catalyst, then we get a very high yield. So there's a dramatic effect of addition of silver in this reaction. And we can see that what happens when you add silver, that the ratio chloro to palladium goes down, and also the XPS is changing from the black to the red. And we believe that the following is happening, that silver is removing the chlorides on palladium to give an active cationic palladium catalyst. Now, this catalyst, which works for a number of substrates of this type, can be recycled. You see many times here without any change of activity. And it's interesting to see in this XPS, black and red. This is after the first run black and after the 9th run red. There is no change at all. So it's exactly the same catalyst. Also the electron microscopy, the TEM here shows after the first and second run, 9th run, they look very similar. So there is no, the catalyst is kept through the reaction. Now, the goal, as Belén mentioned before, the goal is to replace now, let's say, second and third raw metals with first raw metals. And this shows how we have used nanocopper immobilized on microcrystalline cellulose. And microcrystalline cellulose is a very inexpensive material. It's called Avicel, and it has a very low price. And abundantly available, high surface area, biodegradable. And this is where we use this aminopropyl linker because we need this amino group to tie the metal to the carrier. And here is how the catalyst looks like now when we have bound copper. It's dark blue, black precipitate. And this now is shown now for the first time a carbocyclization with the first raw metal nanocopper. And you can see that it works fine. We can have high yields of these reactions. And it's very efficient carbocyclization. It's a very robust catalyst on MCC, and it can be recycled at least more than 5 times, and there is no leaching of copper whatsoever. So we have less than 1 ppm of copper when we check. Now, nanocopper, the same catalyst, has also been used for cross-coupling of propagylic substrates. And in this reaction, you use a Grignard reagent, and it reacts with this substrate, and it replaces this leaving group, mesylate, to give these allenes. And this is now a stereospecific reaction where the Grignard replaces the leaving group with empty stereochemistry. And for those who like the details of these reactions, we generate the copper cuprate-like species from this from this nanocopper, and it reacts now in an empty fashion to replace the mesylate. So the copper is here, and then the reductive elimination with retention gives the allene. So if we start now from optically active materials, which we prepare from enzyme kinetic resolution, and more recently we also work with dynamic kinetic resolution for that, then we obtain enantiomerically pure allenes. And this is an important achievement because these allenes are useful substrates in a number of cases, and some of them are even biologically active. And we have many examples of this. And as you can see, we get very good results here with more than 99% enantiomeric excess and high yields. And we can also have a substituent on the acetylene. And in this way, we obtain this type of now more substituted allenes, also enantiomerically pure. And only given a few examples, we have 22 examples of this type of products. And we have been able to apply these reactions now for the synthesis of natural products. And we have 5 examples of the synthesis of natural products of this type. And one of the examples is shown here. The starting material was prepared from an enzymatic reaction, and then we run with this Grignard reagent, and we obtain this synthetic intermediate, an antimerically pure, and in the end, we have obtained this major antifungal of Sapium japonicum is shown here, a Japanese tallow tree. So that works nice. Now we also have done electrochemistry, and in this case, of course, we can avoid metal catalyst sometimes, and we can have less hazardous reagents, like we don't need to add an oxidant. I'll give you some example here where we have one of our palladium catalyzed reactions, but now we reoxidizing the palladium via anodic oxidation, and on the cathode we reduce hydrogen to protons, and we feed in CO, as I said. And in this way we can run one in an undivided cell, which is unique for such a complicated reaction. And I should mention, you see we get good yields, and I should mention that we can also think of potentially using CO2 as the start. I mean, as the source for CO, as discussed by Belén, because this is a joint project that we have together now. If you use CO2 and reduce that on the cathode with another cathode than the platinum, then we have not obtained these high yields. We have about 20% at most, but this is ongoing research. Then here is just the scope of this reaction. You see we can have a number of substrates, and we get good yields for this. Now, Belén and I would like to thank all the people who have appeared on the slides for their contributions, and we also acknowledge partial support from the Swedish Research Council and the Wallenberg Foundation, the WISE, and in addition to the SafeChem heavy support. So with that, I would like to introduce the next speaker, who is Per-Olof Syrén, and I leave the floor to you, Per-Olof.
Hello, everyone. It's a pleasure to be here. I'm Per-Olof Syrén, and I'm an associate professor in chemistry at KTH. So it's my pleasure to talk today about enzyme catalysis and what we have done in Mistra SafeChem project, and some future directions that I see in the field. Okay. So there is, we are talking about green chemistry here, and we need a lot of green chemistry. So one of the application areas that we have focused on in phase 1 of Mistra SafeChem is actually building blocks for material science, because this is a very important sector, as currently about circa 400 million tons of petroleum is being directed towards making actually synthetic polymers and building blocks for those. And only about 2% of these building blocks comes from renewable sources at present. And synthetic polymers are used everywhere in society, with applications in electronics, textiles is the case we have looked into. So there is a great need to develop tools that will allow us to actually, start from biomass and upcycle such building blocks that are sustainable with sustainable green chemistry into advanced functional materials. And I'm going to talk a little bit about that later in the presentation. One thing to highlight, and I think this is a very important message in Mistra SafeChem, is just because it's bio doesn't mean it's necessarily good. Okay, So we need a toolbox that you know, other people and us have developed in the Mistra SafeChem project to evaluate the processes and the building blocks that we develop, even if they are bio-based. Are they safe? Are they hazardous, etc.? Are they better than petroleum-based chemistry? So we are focusing on using enzymes in green chemistry applications, and we think that that's very cool. They are very interesting catalysts and have a high potential to be implemented in processes that actually fulfill the 12 principles of green chemistry that you see here to the left, taken from ACS. So enzymes are they operate under mild conditions, and they allow us to potentially to enable high activity concomitant with high selectivity and stereospecificity. So there are some ways to work with the enzymes and biological systems to make chemicals. One way is you know, use materials such as you know, cells from plants, and we try to replicate, to do exactly what the petroleum chemical industry does today. We just don't start from oil, but we start from biomass instead. We follow a different approach in my group. So we actually adapt synthetic processes directly to molecules that are abundant in biomass, such as forestry residues, for example. And our aim is to develop processes that are harmonized with nature and adapted not to the petroleum industry, but rather to what nature does. And then we can develop materials that have a different structure than present oil-based materials, but they have similar properties, and they can show, for example, recyclability, degradability, because they are, you know, they are nature-based materials. There are challenges with this, of course. This is not easy, so we need to develop new kind of reaction schemes to upcycle such biomass products. This is one example where we developed actually as part of Mistra SafeChem. We developed, you know, we took pinene from the forest industry, and we developed a chemical process concomitant with enzymes that allowed us to make polyesters such as these, which harbor cyclic motifs as part of their backbones. And we actually have found that this family of novel polyesters show a great potential actually in replacing some of the oil-based materials that we see today. So challenge, besides from scientific challenge, you know, we are actually competing here with industrial processes that are already established, such as, you know, already existing synthesis of polypropylene. So that is one challenge that needs to be overcome. So this is an example of tools we have developed in Mistra SafeChem to actually enable this. So we have developed oxidative chemistries concomitant with enzymology to actually allow us to take from the same inert building block from biomass. We can now oxidize this molecule into, for example, a lactone that allows us to make ring opening polymerization to yield, you know, polyesters. We can also make oxidative cleavage of this ring and end up with diols that then can be copolymerized with actually biomass derived fatty acids and diacids to make very interesting copolymers, such as the ones that are being shown here. This is example of materials that we developed in SafeChem. We can actually make you know, bio-based polymers that we can now assemble on solid surfaces such as cardboard or glass. Okay, and this is controlled polymerization. We can actually, although these are quite bulky residues, we can control polymerization, and we can achieve, well, we can actually control the molecular weight. We can make small, short polymers if we want. We can make long polymers. So this is a controlled process. And these are coatings from nature. I want to highlight that. So this is really interesting. Okay, and when we talk about enzymes for green chemistry, we have some challenges here, because imagine now, typically what we did in chemistry before, we want to make an interesting cool molecule, such as this is taxadiene. It's an anti-cancer molecule. It's actually the precursor of Taxol, so it's not the final drug that I show here. But it's traditionally being isolated from the Taxus tree. And to do that, you needed to basically chop down a 100-year-old tree and extract the molecule from the tree, and one tree was sufficient to make one dose for a cancer patient. And that's not green chemistry. So what we can do today with synthetic biology, we can actually sequence, we can take out the genome, determine the genome from this plant. By bioinformatics, we can understand the genes that underlie the biosynthesis. We can then take out these genes and put them inside the bacteria. That makes a cell factory that makes the same molecule for us. That's pretty neat. There are significant challenges involved in this for industrial implementation. We're talking about stability issues, pH issues, different environments, etc. So in fact, if we take an enzyme from a natural source and we want to plug it into, another cell factor, we want to use an enzyme that makes new chemistry that it doesn't do in nature, we typically observe very low yields. So this is one example. This is some earlier work that we did. This is some very interesting carbon-carbon bond formation processes with terpene cyclase enzymes. And this enzyme is actually capable of making a plethora of different oxidized multicyclic scaffolds. But what we see typically very low yields for unnatural chemistries. Okay? So we need to engineer enzymes to make them better, to make them more stable, to tolerate industrial conditions, and to make them active. And how this is done? Well, it used to be done by structural-based approaches, where one tries to model and understand what's going on. So we took a different approach actually in the Mistra SafeChem project, and we developed a sequence-based protein engineering pipeline, where we actually utilized the vast amount of sequence available, from genome sequencing project. And from that massive amount of data, we can understand actually patterns in genes from homologous organisms, and we can use that knowledge to our favor to actually, from sequence base, without the need for a structure, generate new enzymes that have better properties than those existing actually in nature. So it's something like a machine learning algorithm where we learn from existing sequence based and then use this knowledge to actually gain crucial information on how to improve the enzyme. Some example of work we developed in Mistra SafeChem is to generate, universal pipeline to generate stable enzymes. So we can see one example here. This is some scientific data. It's called a nano-DSF experiment where we can study protein unfolding. Here we start with the wild type sequence in black here, and we generated actually in this case 3 new sequences of that enzyme from sequence space. And by just analyzing 3 new sequences, we were able to find an enzyme that had circa 40 degrees enhanced thermostability, which is quite remarkable. Now allowing us to perform, reactions at higher temperature if we want, and also allowing us to have this enzyme survive for a longer time in industrial processes. And we can also understand what's going on. So we see here in this structural snapshot where the mutations are that we introduce. So we actually introduce significant amount of mutations on the surface of the enzyme. And we can now understand more scientifically what is going on. So in this particular case, we actually by this, we didn't know that when we engineered this, right? But we used sequence information, and after this successful project, we understood that actually a key phenomena that we affected was the motion of this loop here that I show in this picture that is capping the active site. And for the wild type enzyme at high temperature, this loop collapses over the active site and makes the enzyme inactive. But for our engineered variants, it remains open. So we think this is a key process in thermal adaptation. So now this allows us also as part of the Mistra SafeChem project to engineer, we can use this knowledge that we gain to actually generate not only highly stable enzymes, but also enzymes that are highly active and specific from, combining sequence-based engineering with actually more rational approaches, which is illuminated here. So it's quite interesting to see that we not only can make stable enzymes, but also enzymes that are active. Then we also developed an approach using this pipeline to make also bioplastics, not only in milligram scale, but in a little, let's say, more preparative scale. So this is FDCA, which you might know, it's a key bioplastic precursor that is of high interest. Traditionally, it is made by this C6 route, starting from cellulose. And then you go over a glucose to fructose isomerization step, which is quite actually challenging. And you have HMF here as a key intermediate. HMF is unfortunately quite toxic to cells. So we were thinking if maybe we can develop a C5 based route, so starting from 5 carbon sugars, and then actually make a key carboxylation step here that can actually allow us to make FDCA by a CO2 sequestration process. And this works, and we actually developed a cell factory for this process, which is based on normal gut bacteria supplemented with a carboxylase enzyme. So we can now start with C5 building blocks. We can carboxylate them using CO2 to make FDCA. And it's actually based on quite complex biochemistry, which is dependent on these prenulated flavin cofactors, which is shown here. And they are in fact light sensitive. So before, when people worked with these processes, you actually had to purify the enzyme and then wrap aluminium foil around the tube, so to prevent this actually cofactor from degrade. And that doesn't work in industry. So we actually developed, 2 minutes, I see. So we actually developed a process we were thinking like this, that what if we take E. coli cells, and which actually have the cofactor maturation system to make this cofactor, and we supplement it with this external carboxylase. And in that case, this actually worked. So now we have a cell that protects the enzyme from light and oxygen, and we don't have to purify anything. So we can use the whole cell as catalyst. And it looks like this, this is the PhD student actually making FDCA in the lab. So this works really neat. Was also got quite a bit of attention, and you can see here from HPLC traces that this works quite well. We get already after just only 3 hours, we have already a significant conversion without any optimization of the process. This is NMR data confirming the product, the integrity of the bio-based FDCA that we made in our cell. It is also compatible with a lot of furan derivatives, so it works, for example, with purols, which is quite neat. So they also seem to be able to enter our cell factory and becomes carboxylated. So this opens up quite interesting doors now to make, structure functional relationship studies and to make, new kinds of biopolymers. It is even possible to use an enzyme to make these polymers. This is not the green process. I mean, you see toluene here, so this is not really green chemistry, but it's cool to see that an enzyme can accept these building blocks that were made by an enzyme to make an enzymatically made polymer with high molecular weight. This is the polymer that the PhD student made from this process, and in quite high molecular weight, over 100,000 grams per mole. I would like to close by saying that in the final stage of the process, we actually developed a safe by design approach in enzymatic synthesis of amide bonds, where we actually strive to incorporate the different nice research that's going on in the in silico toolbox work, led by Hanna, Ian, and Peter Fantke and others. And we were actually, we were having the idea that we would like not only to make green chemistry, but we would like to make green chemistry that's safe. So we developed a process where we actually, incorporate the in silico toolbox from Mistra SafeChem, allowing us to filter out building blocks already before a synthesis is being made to generate building blocks that are safe. And we call these building blocks SafeChems. They are safe chemicals that can be manipulated by enzymes. And we can develop enzymes that can, couple this kind of SafeChem together in coupling reactions, which is seen here. This was a nice collaboration, with AstraZeneca, where a PhD student of mine, Elizabeth, was there to do this nice work, where we actually not only can, filter out safe chemicals, but we can also couple them together to make very interesting products. And for final slide, I would like to say that we also incorporate use tox developed by Peter Fantke to benchmark this safe by design process that we are striving to develop. And it seems like this filtering process works quite nicely in terms of safety. And now this allows us to move towards, fate and exposure, and not only hazardness actually. So very important to develop safe chemicals in the Mistra project. So with that, I would like to thank you so much, and thank you so much everyone, contributing to this. Elisabeth, my PhD student. We have nice collaborations within VP4, and especially I would like to highlight the collaboration we have with Ian, Hanna, and Peter Fantke in the project, which is crucial, and with AstraZeneca, of course, Magnus and Martin. With that, we can move to the next speaker, which is Professor Mathew from Stockholm University.
Hello, everybody. I'm Mathew, professor in material chemistry from Stockholm University. I will continue with the next part of the talk, which is towards safe and sustainable upcycling of forest and textile residues, and I will give an overview of this topic. And this is a cooperation with the Joseph Samux group at Stockholm University, and I will be covering both the presentations today. And this will be also continued with the presentation from two of our PhD students, where you will hear much more about the scientific part of this work, whereas I will try to give an overview of the concept we have developed and the approach we have taken in this project. Moving on, so the components here was to have green and sustainable chemistry with respect to the topic we have been working on, and we have been focusing on these three aspects, basically materials and chemical building blocks which are safe and sustainable, and how to make new industrial and consumer products from this kind of materials and chemicals obtained, and also see how much we can use or reuse resources which are around to make this kind of materials, and at the same time make sure that these processes and the products are safe and sustainable. And this was the basic concept we want to have in this project, and here this is the focus of the project. So this was about recycling of textiles, which is the post-consumer textiles, as well as forest residues, which is not currently used, and use them to produce chemicals, and as well as material. Here is nanocellulose, which is a cellulose in nanoscale. And what we did is we developed scalable fractionation routes to generate useful chemicals and nanocellulose, and also make new materials from these recycled products. And another focus of this work was to optimize and characterize these materials, and also to see how safe it is during the processing. And this was supported by life cycle evaluation, and also evaluating the toxicity of the processes, intermediates, and products. And here in the next slide, you can see a little bit how we have approached this one. And this is the overview flow chart of what was done in the project. So if you take the two raw materials we have worked with, which is recycled textiles and also forest residues, we developed different fractionation routes, as you can see here, and we fractionated into different fraction, including chemical blocks, which is more chemical based, and then also the cellulose, because both the sources have cellulose as a common fraction. And the cellulose is used to make nanocellulose. For example, in the case of recycled textiles, nanocellulose means both crystalline regions of nanocellulose and as well as nanofibers, and then they are further used to make new functional materials. The same with the other side, like we started to make cellulose, and then we go into fiber spinning to make different kind of nanofibers, and then we have achieved those also. In addition to that, we get different chemicals. So this was the overview we have taken, and also at the same time we were always trying to see chemical tracking during the processes, like example dyes, for in the case of textiles we were tracking, and here we had support from the analytical chemistry group from Ulrika and Nielsen to take a look at that part. And moving on, so we also have taken a lot of interest in collaborating with other work packages in this project, which is work package 3, where we did all the toxicity screening and comparison of the analysis of forest residues and also the textiles. Then we also had life cycle analysis of these systems evaluated in work package 5. So this was very collaborative effort, and this is also something we learned by doing, how to collaborate with other work packages, other outlook in while we are making the materials and the chemicals, and also developing recycling techniques. And I would like to take one example here where we did a screening life cycle analysis, which is from cradle to gate approach for a laboratory scale process we have developed. This is on cellulose nanocrystals prepared from textiles. But here we compared that with the cellulose nanocrystals prepared from wood pulp. And we used a sulfuric acid hydrolysis process as a standard method, and also a citric acid hydrolysis process, which is a more new method developed under Mistra SafeChem. So here from the screening LCA, which was done in collaboration with Rice, Anna Karin and team. So we could see that the processing of cellulose nanocrystal from post-consumer cotton by sulfuric acid hydrolysis, which is shown by the light blue color bars, is actually much more environmental friendly than wood pulp as a resource. But then when we come to citric acid system, it is not the case. It is almost the same as the wood pulp. And you can easily see that the eutrophication is one of the most key hotspot in this screening LCA. And when we analyze the hotspot more clearly, it is possible to see that the hydrolysis is the most dominating process, and the main contributor is the citric acid itself. So we found a way to mitigate that by recycling the citric acid in the process, and then we could see that we could recover almost 60% of the citric acid from the effluent, and also we got it with a high purity so that we can use it back in the process. And this is a little bit the approach we have been taking, so that we can use the life cycle analysis or the toxicity screening information to improve our process and almost optimize the process for more safety and sustainability. And I would move on to say that there have been several people who has been involved in this project. And Suthawan Muangmeesri is the first one, and is a PhD, and also Maria Jimena, and both of them will be giving the presentation after me today. And then we also had some other people involved in the project, both postdocs and master students. And in summary, I would like to say that this project, we have developed new processes to valorize forestry residues and post-consumer textiles, where in tandem with chemical and environmental safety consideration. And we have separated textiles into components and also forestry residues into different components. And we also did the life cycle analysis, and we also did the toxicity evaluation of the products. And what is important and key to this project is this multidisciplinary approach, where we have a material chemistry, organic chemistry, analytical chemistry, life cycle analysis, etc. coming together. And in this project, we have approximately published 15 papers as of now, and some including cover arts. And with that, I would like to leave the word to the next person, which is Suthawan Muangmeesri.
Thank you very much, Aji. I'm Suthawan Muangmeesri, a PhD candidate at the Department of Organic Chemistry, Stockholm University, under the supervision of Professor Joseph Samek. Today, I'd like to share some of the recent research that have been conducted and are currently ongoing in Joseph Samek labs under the Mistra SafeChem funding. While there are several exciting projects underway, I'll be focusing on two key studies that are particularly noteworthy. The first project, catalytic fractionation of spruce bark. In the forest industry, wood needs to be disintegrated for further use. It can be divided into two parts, 70% saw logs and 30% forest residues. The saw log, which make up 70%, are widely utilized in pulp mills and sawmills. However, the remaining 30% is considered low value and is currently burned for energy. Notably, 50% of this residue is composed of bark, the outer layer of the tree. Our initiative aims to valorize this low-value residue into high-value cellulose pulp and chemicals via the lignin-first method, oxidative catalytic fractionation, or OCF. While wood has been extensively researched, bark remains less understood due to its complex component. Notable studies include Christo Coppola's use of organosulf pretreatment for cellulose and Monica EF's extraction methods for lignin. Our work adopts a holistic approach using the lignin-first method, OCF, to fractionate bark, upgrade cellulose to nanocellulose, and depolymerize lignin to high-yield monophenols. We also explore new uses for lignin in bark. Bark is underexplored and less understood compared to wood, with limited knowledge about its lignin typically assumed to be linear. We achieve a high yield of lignin monophenols, notably vanillin from bark. Using 2D NMR, we explore the lignin structure and obtain valuable insights. Our analysis suggests that lignin in bark is cyclic, aligning with our findings. We demonstrated the effectiveness of oxidative catalytic fractionation, OCF, in upgrading cellulose to nanocellulose and depolymerized lignin to high-yield monophenols. We also showcase the mass balance of our process to ensure minimal loss and optimal resource utilization. In summary, our project involved upgrading lignin to higher value monophenol products while preserving other constituents, alongside upgrading pulp to nanocellulose. Additionally, we propose a new model of bark lignin correlated with observed high monophenol yields. And the second project, polyester depolymerization. PET, or polyethylene terephthalate, is a widely used thermoplastic, creates a substantial non-degradable waste, adding to plastic pollution. As demand grows, it's urgent to find a sustainable way to manage PET and curb future pollution. Varied routes exist for recycling PET into its building blocks, including hydrolysis, glycolysis, and alcoholysis, each with its own advantages and drawbacks. In our research, we focus on organocatalyzed methanolysis, enabling solvent mixture circulation with a reduced solvent to substrate ratio. We conduct a life cycle assessment to evaluate its environmental impact. We demonstrated the capability of our developed system to recirculate the solvent up to 5 times. Additionally, we collaborated with KU Leuven to conduct a comprehensive life cycle assessment analysis. We extended our research to polycotton fabrics, recognizing its common composition of both cellulose and PET. Our findings reveal complete depolymerization of PET alongside isolated cotton. Our process efficiently converts PET to dimethyl terephthalate and ethylene glycol with a recyclable solvent system, achieving high purity and recovering 90% of solvents. The environmental impact is significant, with a negative 1.88 kilogram carbon dioxide footprint, offering a sustainable solution applicable to various polyester and polycotton waste streams. Last but not least, I would like to thank my supervisor, Professor Joseph Samek, and my co-supervisor, Professor Aji Mathew, for the support, and of course, the Mistra SafeChem for their funding. And with this, I would like to present the next speaker, Maria.
Okay. Hello, everyone. My name is Maria. I'm a PhD candidate at the Stockholm University, and I'm under the supervision of Aji Mathew. She recently presented my work, some highlights, so I'm just going to dig more into that. Then then I will just start to show my slides right now. So then the title of my project is Valorization of Post-Consumer Cotton Fabrics into Cellulose Nanomaterials and Their Applications. And the first thing that I want to do is to explain what we are doing in this job. So all of us wear clothes. I mean, they have been with us forever, and who doesn't like to buy new clothes? But the problem of this is that we don't know how to properly discard them. So if we look at the market, we actually have 3 fabrics here, 3 types of fibers. The most common is polyester. The second one is acrylics in countries like Sweden, which gives like a wool feeling, a wool type of feeling. And the most popular natural fiber, and that has a consumption of 1/4 of it, is cotton. Cotton is mostly made of cellulose, and this is natural. And with the finishes that you do during the textile, actually the content of cellulose goes up to 99%. So the idea of my work is try to take advantage of this high cellulosic content. If we look more into detail into these fibers, we can see that the cotton fibers, I'm pretty sure that all of you are familiar with this picture. When you have done laundry and try to clean it up, you see these little fibers. These fibers actually are composed of something called elementary fibrils. So these elementary fibrils are arrangements of cellulose chains that they are hierarchically arranged into the walls of the fibers. Then if we are managed to isolate these elementary fibrils, which are in the order of nanoscale, we can have a nanomaterial with a very light aspect ratio. Now, if we take these elementary fibrils and through a chemical process or something, we manage to cut them in certain locations, then we can isolate rod-type nanoparticle cellulose nanocrystals. And my work during these last 4 years has been to extract these nanofibrils of nanocrystals from cotton. These 2 are called in general cellulose nanomaterials, and the purpose is to see if we can actually, instead of using virgin sources, use use discarded textiles as a source of cellulose nanomaterials or CNMs. So here I'm just going to give a very quick detail of how these routes are done. So then we start with textiles with cotton, and then through a sulfuric acid hydrolysis, this will be like the scissors that I showed in the picture before that will help us to isolate these rod-type particles, and we also will have a surface charge that help us to give some extra properties such as colloidal stability. We also try different routes. So then we used a chemical process to give the colloidal stability, and here to a mechanical process, we made the scissoring from this part in order to isolate these rod peaks, but instead of having sulfate half ester groups, we will have these citrate groups that will give us other type of applications. And then we actually try another route that would allow us to also use a strong oxidizing agent in order to have particles with a lower percentage of impurities. So then these 3 types of nanoparticles are the ones that I firstly extract from textiles. It's important to point out that we didn't do any cleaning or anything. We just took as received textiles, and then we just processed them as I previously explained before. If we look into detail to the morphology, we can see these rod-like structures that I previously shown. And we can see that they have different changes in the morphology depending on the route that we use. So then we can tailor the properties of these materials. If we look a little bit about the surface charge, this is a very important parameter when it comes to the application of these materials. We can find that they are in the same order of virgin sources. The second type of material that I explained before, it was CNFs. So we also take these cotton garments. And then the first thing that we do is to give a surface charge. Here we aim for 2 different types of charge. So in the bottom, in the upper part, we have a negatively charged compound, and in the lower part, we have a positive one. And then through a mechanical fibrillation, we manage to isolate these long aspect ratio particles that I mentioned before. If we look more into detail about these materials, then we can see them first that since they are so long, they form gels. And what is interesting to see here is that in the upper part, we don't see any part of the dye that we had before. In the lower one, we can see a little haze, but still it's a very clean and pure material. If we look more into detail into its morphology, you can actually see these nanoparticles in the nanoscale. And we can see that depending on the route that we use to extract them, they have different morphologies. And finally, what is important for this is the surface charge. As I mentioned before, this will help us to have different applications of these materials. And the numbers that we got, they are similar to the ones getting with virgin sources. Here I'm just going to briefly show an application. The second part of my PhD has been to focus to give different applications to these materials. And then the first application that I want to mention is a lightweight material. So a lightweight material is something whose apparent volume is way bigger than the volume occupied by the cellulose. So this is a material that is pretty much air. And one of the materials that we formed from this, it was a cryogel. So then how these materials are done. So we take a dispersions of either the CNCs or the CNFs that I showed before. We do something called an unidirectional freeze casting. That the idea is that we will use ice as a template in order to make growth these materials in a specific location. And then here I just show a little more into detail. So then when the ice is growing, this will help us to get some walls in our. They will help us to make a template of our materials. And then we end up something that is an anisotropic material. And what is the reason that we want anisotropy is pretty much because we can tailor the properties of those materials. So we took the nanoparticles that I showed you before, and then we end up with this material, this foam, that we characterize its porosity. If we look more into detail into this morphology, we can actually see that in the axial direction, you have these columns which are given by the template of the ice, and in top we can see a cross section where we can see elongated pores, which is also due to this templating technique. These are very light materials, as you can see before. Like we can just put them to give a visual of how they are. And the purpose of these materials is that we can use them as an isolating agents and other parts. And well, I went faster than what I thought, but then now I want to introduce for the Q&A session, and I just want to acknowledge my supervisor and of course Mistra SafeChem for funding my project.